You know, you may hear it said that old Professor Allner, he's got a lot of nerve. That is not a description of my personality. It's quite literal. I have a bunch of them. You may also hear it said that we A&P nerds have a lot of brains. Well, no, we're smart and we have a lot of brains. But-um-bump. Yes, but-um-bump indeed, everybody. Welcome back to Bio 223 Lab, right here at the West Charleston campus, with me, Professor Allner. Am I getting a little goofy being in lab all this time by myself? Well, that's really just a convenient excuse, because guess what? I was goofy before all this. Welcome back to lab number 7. So lab number 7 is the Neuron and Brain Lab. This lab, right here, purely anatomical, meaning you are going to name the parts, and then you're going to name some more parts, then you're going to name some more parts after that. What we have is the anatomy of a neuron, its cell. Yes, in my gag before I said nerve. Those things are not actually nerves, they are single individual neurons, and we will take a look at the parts of these cells. Then we have brain anatomy using those models I showed you over there, naming a whole bunch of parts. Then we have more brain anatomy with the sheep, using an actual sheep brain dissection. Why sheep? Simply put, sheep are cheap. We can get sheep brains and I can dissect them for you. I got to fish them out of a bucket and then show you some of the parts. Sheep are also mammals, so many of these parts will be the same. Now, looking at these, as I mentioned in the intro, we always want to look at the models first, then the actual dissection. Hopefully it won't be that bad for us, but there is one problem to overcome. That problem goes something like this. And do not attempt to zoom in with your streaming. You'll get to see this close up. But a typical problem challenge in lab number 7, watch the tip of my probe here. So I'm pointing at the hypothalamus, the infundibulum, the pituitary gland, the intermediate mass, the fornix, the anterior commissure, and the pineal gland, all within an area less than 1 square inch on this model. My point is this: these are small, very discrete structures. Oftentimes during this lab, when students are here in person, it's mostly me or your lab instructor walking around with a probe just pointing at things. You know, where is this? There it is. Where is this? There it is. And many of these things are very close together, so it's hard to tell. The beauty of an online lab, and I'm not trying to sell you on online labs, but one benefit you get is I can zoom in with this camera right here and create as high quality an image as I am able that shows you exactly where these are. On your quiz or your practical, I can put a dot exactly where I want it to be, not a piece of tape that I slap on there. So it should be fairly easier for you to memorize these parts as we go through them. But for this lab, this instructional video, you would, if you are wise, watch it a number of times and really be hitting that practice video hard, because this is all naming of brain parts. What's this part? What's that part? And drill and practice is the way to go. And to help you remember what I said in the intro, use this set of diagrams that you have here. They are extremely helpful. Let's begin our memorization in lab number 7 with this thing right here, which is a neuron. So this is the first section of lab number 7. Find it on page 1. certain parts of these cells that you must know. Now, I dug out this rather old neuron model because it has a couple very nice things that I would like to show you. So keep in mind that what I'm talking about is one individual cell of the nervous system, a neuron. This one is quite a bit enlarged, but not as long as some. Everybody watch this. This is a meter stick I just set next to this thing. And this particular model, the neuron is maybe 68 centimeters long. I could probably dig one out of many adults that would be 1 meter in length. One individual cell in a human's body that's 1 meter in length. So they get the title of the body's longest cells. And don't even get me as a biology nerd started on how long some of these things could be in other critters. Say a giraffe, an elephant, a blue whale, a giant squid that's 60 feet long. So neurons can be quite long, but this one's been greatly expanded and magnified so we can see some of the general parts. Now, as we look at this thing, what we can see right now, first off, right here, this large section of the cell where the nucleus is located, this is the cell body or the soma. We will say cell body for your quiz and your practical. So this is the cell body of the neuron. Then we have projections coming off the cell body. As you may have already learned in your lecture, the number of projections dictates what style of neuron we have. This one has 1, 2, 3, 4, 5 projections off the cell body, which makes it a multipolar neuron. But for our lab purposes, all we have to do is name some of these projections. So again, the cell body is right here. These things, these long, branchy green things, are the dendrites. So dendrite, 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 branching off of the cell body. Dender makes reference to branching like a tree. Then we have what essentially amounts to be the long biological wire right here called the axon. This is the axon. So this long projection that comes off the neuron and extends is the axon. And what you can see right here, follow my probe around and we will zoom in a little bit, where the cell body narrows down to become the axon, we have a small, in 2 dimensions, it's a triangular looking bit of the cell. In 3 dimensions, it's actually a cone. But right here where the cell body narrows down to become the axon is what's called the axon hillock. You'll hear terms in your lecture like initial segment and trigger zone related here, but for lab, axon hillock is right there. Now let's move in a little closer on this particular neuron to examine that cell body and a couple of the structures we see there. So here we're zoomed in a little bit on the cell body of this particular neuron. I can see the nucleus right there, which is not on your list. But 2 things that are visible here are on your list: the Nissl bodies and the neurofibrils. Now, I only say this to you people because, think back to my earlier gag, you have a lot of brains, but these cells have a huge amount of endoplasmic reticulum with what amounts to be these very large ribosomes on them. Look at all these Little red, they look like little beads or spheres here. These things are Nissl bodies involved with the manufacture of proteins because these cells make huge numbers of proteins, aka neurotransmitters. Not that I want to get all lectury on you here. So these round red spheres are Nissl bodies on this model. The long strands of red that we see here look sort of like, red, tiny, thin licorice or something. These are neurofibrils. The neurofibrils help transport the proteins around. So, cell body, Nissl bodies, and neurofibrils in this particular neuron. We're going to use other models to see some of the other features because they show up a little better on them. So here we see another neuron model that I like quite a bit for a few other reasons. So we can see right here this large cell body with dendrites coming off of it. I can even see lots of other neurons addressing this one, lots of neuro-neuro junctions here, but these big things right here are the dendrites, but they've been cut off sort of short. I can remove this part of the cell, and we can see all that endoplasmic reticulum in there and all these ribosomes, but the Nissl bodies aren't as good in this one as they are in the other one that we saw. I can definitely see a very nice axon hillock right here, which is even shaded a different color for us on this model, leading us to the axon, this very long projection of the cell that travels so far in many of these neurons. But I do have to point out a naming thing that will probably irritate you, just like it irritates me, but nobody considered our irritation before they named this thing. Watch my probe. Right here. Right here. This sort of light gray material. Right here. What is that? That is the cell membrane of the neuron. See this sort of light yellow business right here? This is another cell, an accessory cell to this neuron. This thing right here, from here to here, is a Schwann cell. The cell that wraps around and around and around the axon to, in effect, in a biological sense, insulate the wire so the action potential can down this thing using saltatory conduction. Again, getting a little lectury on you sometimes is hard, sorry. But this cell is what's called a Schwann cell. Look for that on your list. So there's a Schwann cell right there. Here's another Schwann cell right there. Notice how each Schwann cell has its own nucleus because it's a different cell. Now, here's the irritating part. Watch my probe. What am I touching right now? The cell membrane of the Schwann cell. What's this called? The neurolemma. Do you see that in your list? The bottom of the left-hand side. The neurolemma. Wouldn't that name make you think that it's the cell membrane of the neuron? Au contraire, I say à la française, and that's for a reason. This is called the neurolemma, even though it's the cell membrane of the Schwann cell. This, the light gray part, is the cell membrane of the neuron, which is in this area surrounded by these Schwann cells. This is actually called, and I say this just because I know some of you want to hear it, this is the axolemma or the axonolemma, which is not on your list. So for our list, we have the axon hillock right here. The axon right here, which you can see they sort of opened up for us so we can see inside it. And then Schwann cell, Schwann cell. On the far right of the image, as I move this over, do you notice this connective tissue right here? Always get people trying to learn this who think this is the neurolemma. No, this is endoneurium, one of the connective tissue elements that surrounds a nerve, not on our list either. So here's a Schwann cell right there, here's another Schwann cell right there. One Schwann cell, two Schwann cells, and part of a third. Neurolemma, axon, axon hillock, way up there. This particular cell does not have much in the way of good neurofibrils or Nissl bodies, but it does show me this covering very well, right here. Now, I want to be very, very clear with you because you are going to be taking a two-dimensional quiz and practical over this thing. Let's be careful with our terminology. So do you notice where one Schwann cell ends and the next one begins? I can see it right here. I can see it right there. There are actually small microscopic gaps between these two cells. The gap between one Schwann cell and the next is called a node. These are our nodes. Look for this term in your lab manual, the nodes of, and then you have that R word. This is one of the few places where we still have people names on things. What's this called? The node of, and do not do the American thing and say a word that rhymes with reindeer. That's à la française. See my French reference before? If I was Dr. Besson here, I would have no problem with the pronunciation. But how would you pronounce that R word à la française? Ranvier. So this is a node of Ranvier. This is a node of Ranvier. The gaps between the Schwann cells, the nodes. Many Americans just say nodes. They don't even try to pronounce the word. What else I can see is this idea. Again, be very careful because it can be confusing. We have a structure called a myelin sheath on these cells. It's in your list, myelin sheath. The myelin sheath is the collection of all the Schwann cells together. Now, what have you people noticed in these online labs? Oh yes, everyone knows. Professor Allner is quite a fashion presence. Avant-garde, fashion forward, that's Professor Allner, isn't it? I say that in jest, but I want you to think of the outfit you just saw me wearing. Didn't I have a shirt? Didn't I have pants? Didn't I have shoes like that? So the combination of all my clothes together, shirt, pants, shoes, belt, etc., that's my outfit or my ensemble. My shirt is just my shirt. My shirt is not my outfit. My point is this. Here's a Schwann cell. That's not the myelin sheath, that's a Schwann cell. This Schwann cell plus this Schwann cell plus this Schwann cell, that is a myelin sheath. So on a quiz or a practical, if I had a dot right here and I said name this cell Schwann cell. If I had a dot right here and I said name this structure node of Ranvier. But if I had dot, 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 dot like that, multiple dots across more than one Schwann cell, that's got to be myelin sheath. If I turn this model, I think you can see and let me do a little bit of super camera work here. Do you see what these Schwann cells do? I'm going to try to tilt this without actually striking the camera. There we go. They wrap around the axon of this cell multiple times, around and around and around, like paper around a cardboard tube for paper towels, if you can find some paper towels, which they're available now. So, but see all these layers of cell membrane, which is mostly what? Phospholipids. That serves to insulate this cell. So the surrounding of the axon by the Schwann cells, like that, creates what we call the myelin sheath in total, with these nodes, gaps between the Schwann cells. So again, one Schwann cell is a Schwann cell. Its cell membrane is the neurolemma. It surrounds the axon. Multiple Schwann cells together, 2 or more, is the myelin sheath when they're all coiled up and wrapped around the axon like this one. Let's look at one more neuron model and then do a synaptic junction before we leave this anatomy. So here's the last neuron model I wanted to show you. So notice cell body with these nice dendrites coming off. And again, this one is trying to show me parts of a whole bunch of other neurons that are connecting with this one here on its dendrites. But let's remove this part of the cell, turn it over. Look at these beautiful sort of brownish black neurofibrils. Look at these beautiful blue, purple, gray, blue-gray Nissl bodies. Axon hillock, axon, Schwann cell, neurolemma, node of Ranvier. Here's that endoneurium again, which is not on our list. Cell body, axon hillock, axon in the middle, Schwann cell, dot, 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 myelin sheath, node of Ranvier, neurolemma. Now we've hit every single structure that's on this list except for a synaptic junction. This neuron's been cut off right here. It would continue to your right. I need to go back to that old model to show you a synaptic junction, so let's do that. Here's that very same older neuron. And as we go down, notice the Schwann cells on this one, while they are present, they're not so hot. And the node of Ranvier right there, it's there, but it's not that great. The other models are far better for that. But look at this. I have a synaptic junction very nicely illustrated on this model. It's the spot right down here where this neuron releases neurotransmitters onto this cell. Now look at this thing. It looks sort of like a piece of jerky. That might be because it's supposed to represent a skeletal muscle fiber. So the synaptic junction is right here where the neuron spits its neurotransmitter onto the cell membrane of this muscle cell right here. For people in one of my lectures, right here where I'm pointing is where happens. That's it right there. That's the spitting sound, where a neuron spits its neurotransmitter onto its target cell. My other 2 models over there don't have this synaptic junction. They had a few on their dendrites, but not like this one. So see that yellow right there? That's right where the neurotransmitter gets released, a small gap from neuron to skeletal muscle fiber where we spit the neurotransmitter across that gap. So feel free to rewind this, play it several times. The anatomy of a neuron. right there. Okay, now let's get started on the anatomy of the human brain. What you see before you right now, actual human brains in a plexiglass case filled with preservative. I like to call these my brains au juice. Brains in juice. Now, I use this to demonstrate some of the things that show up first. Now, when people think of a brain, here's a rubber brain, they think of a brain as a solid mass of cells and tissue. Well, it's mostly solid, but not completely solid. Your brain does have a few hollow spots in it, namely 4 of them. So as you look at first this model of a brain, now I'm not trying to teach you these brain regions right now. We have models for that. I will show you, but we have the brain stem right here, the cerebellum, and the cerebrum, the great big wrinkly thing. And we have hollow spots that live within and between some of these brain regions. I can see the same thing right here. Notice the cerebellum, the lighter colored brain stem because it's mostly wiring. Those myelin sheaths, those Schwann cells are lighter in color. And then the cerebrum. And if I look at this frontal section, so I want you to imagine this brain right here, if I cut it this way, so you were looking straight on into it, do you notice the hollow spots? Right here and right here, where we would see and find cerebrospinal fluid. There's 2 of them right there. There are 4 altogether. This model right here, which is a relief model, imagine if I took those hollow spots and extracted them from your brain, what it would look like. These 2 big things right here are the lateral ventricles. Right in here, if I can get my finger in there, there's the 3rd ventricle and the 4th ventricle with the cerebral aqueduct right there. Now, this model's a little tough to understand. I'm not a big fan of it. I'm not going to use it on your quiz or your practical. I know some of my fellow instructors really love this one, so let me take a quick vote as I do. Anybody in here strongly want the students to be tested over this model on their next quiz or practical? Hearing none, the motion is decided. We will not use that one for your lab or your practical. But we do have these hollow spots. The lateral ventricles, 2 of them. The 3rd ventricle, 1 of them. The 4th ventricle, 1 of those. So a grand total of 2 laterals, the 3rd and the 4th, 4 hollow spots found in the human brain. Let me zoom in on this frontal section right here so you can even see it labeled. So I've got 2 hollow spots, 1 here, 1 here. Those are the lateral ventricles with a little wall between them. That wall is called the septum pellucidum. I want you to look in your lab manual. Remember how, thank you Professor Hicks and Professor Larson, it's numbered, and the diagrams, you got to use those. It's already drawn for you. Lateral ventricle here and here, and the septum pellucidum, clear wall separating them. Now, I'm not going to use this particular thing to ask you these parts on your quiz. Why? Well, duh, they're labeled right here. Oh, certainly I could use the back side if I wanted to, but it doesn't look so good on the back side. and I don't want to go fishing around in there and take the labels off. But I can show you this on some of our brain models. So what we're looking for are these 4 hollow spots in the brain, and look at all the way down at the left side of your column on page 2, the septum pellucidum. Okay, I want you to look at this brain model right here. This is where I would find the lateral ventricle and or the septum pellucidum. Right here, a hollow spot in the brain. When a model maker constructs this thing, they only get 2 choices. They can either show me the septum pellucidum like they did in this half of the brain. There's an actual physical wall right there. Or they could show me the cavity, the depression, which is the lateral ventricle, which is behind the septum pellucidum, right here. So this is that cavity, the lateral ventricle. Most of the models do not show this lateral ventricle. They instead show the septum pellucidum. Let me show you on a few of these. Here's another brain model. Right here is where the lateral ventricle would be, but there's a wall there, septum pellucidum. Here's another brain model. Right here is where the lateral ventricle would be, but there's a wall there, septum pellucidum. I do have one particular model that shows the septum pellucidum quite nicely. This one does, you already saw it. Watch my probe, it goes right in there, that's a cavity, but it's not the greatest. Let's take a look at the single model I have here in lab that shows the best lateral ventricle. Okay, I want you to look at this model right here who's actually, you can see, keeping me company right here. No, he doesn't speak. Let's just call him Wilson. Wilson! Like that. Okay, for the movie buffs. Now, this model has brain, which I can remove and bring in front of you like so. Is there any question in your mind looking at this model that there's a big deep cavity right here? That is one of the 2 lateral ventricles. So if I have a dot right in here, see it even casts a shadow there. Look, my probe goes into it. This is the lateral ventricle. We have 2, one on each side of the brain. You have a left and a right lateral ventricle. You do not have to name them left or right. You just have to name it lateral ventricle. The lateral ventricles are actually numbers 1 and 2. The third ventricle is this cavity right here. So see my lateral ventricle right up here, everyone. This is the third ventricle right here, sort of this shallow depression. Let me angle the brain model a little bit. I don't know if you can see, if there's enough detail. If I had liquid in there right now, it would hold like a shallow pond. This is the third ventricle right here. Imagine when the 2 halves of the brain are together, it's sort of like cupped hands where you leave a gap between your hands, so I could fill that with fluid. That's my hollow spot. So the third ventricle is right here. Lateral ventricle, third ventricle. Let me show you on another brain. Right here. See the depression that the tip of my probe is in right there, this little shallow pond? Third ventricle. One more. Shallow depression right here is the third ventricle. Right here where the tip of my probe is, between the brain parts we call the cerebellum and the brainstem. Actually, the specific part of the brainstem called the pons, right here is where we find the fourth ventricle, between the cerebellum and the pons, connected to the third ventricle by this canal right here. This canal, this tube right here called the cerebral aqueduct. Cerebral aqueduct. Fourth ventricle right there. Third ventricle right here, connected by the cerebral aqueduct. And in a living brain, these hollow areas would be filled with cerebrospinal fluid that would eventually go into the central canal of the spinal cord and fill up the subarachnoid space. That's just a bunch of lecture stuff for you. Beautiful model of what? Lateral ventricle. There's a pretty nice fourth ventricle. The third ventricle here isn't so good. The only other model I have that shows a lateral ventricle, there's the third, there's the fourth, cerebral aqueduct. Hopefully you see those. Make sure you pay attention to it in the practice video. Now, before I leave these hollow spots in your brain, which you might be surprised to learn your brain has hollow spots in it, but it does. What you need to pay attention to on your quiz is the question that I ask you. In a two-dimensional lab quiz, do you understand that it could be difficult to tell this lateral ventricle from this septum pellucidum, because they exist in the same spot, and in a two-dimensional space, oh, I can turn it and rotate it and try to make this as beautiful as possible for you, but it's still tough to tell. So the question I ask you will dictate your answer. What is the name of the cavity found here? Has to be one of the ventricles. Lateral ventricle. What is the name of the cavity found here? Fourth ventricle. What is the name of the cavity found here? Third ventricle. What is the name of this structure? Septum pellucidum. Did you notice the difference there? So just starting in our top left, we have four, count them, four hollow spots in your brain, two lateral ventricles, a third ventricle, and a fourth ventricle, along with the cerebral aqueduct and the septum pellucidum. Find them on the diagrams and label them. Color them in, highlight them, do whatever it is that you do. Okay, now let's begin looking at some brain structures other than the hollow spots in your brain. And I will be jumping around a little bit between the left and the right-hand columns in your lab manual, but make sure that you're using those, again, very helpful diagrams that have been provided for you. I strongly suggest coloring those things in, labeling those things. We have all the numbered labels and everything that I went over with you in the introduction to this lab. So moving down in the left-hand column, we see the three-parted brain stem from here to here. So if I indicated this entire structure, that's the brain stem. Now what I've done is set up four different brain models so you can just see slightly different views of it. Brain stem right here. Notice how it's shaded lighter because it's primarily just axons of neurons. And here's one in facing the other direction. This is the anterior, this is the posterior. Brain stem right here. And a last one, very nice, right here, brain stem, right there, this entire region, leading into the brain. Now what we see is down here, the spinal cord actually expands out then expands out again, and then narrows up a little bit. We have three parts to this brainstem. From inferior to superior, we have the medulla oblongata right here, the first bulge. Everybody loves to say medulla oblongata. The pons, and then above that, where it's a little narrower here, the midbrain, right here, midbrain. Medulla oblongata, pons, midbrain. Let's look at it on the other models. Medulla oblongata, a little weak here, pons, nice, easy to see, and the midbrain. Notice that the fourth ventricle is directly behind the pons, just as an anatomical frame of reference for you. Medulla, pons, midbrain. And back here to this one, medulla oblongata, the pons, and this whole area right here, I'm circling it with my probe, is the midbrain. Now there are some structures that are associated with the brainstem that you need to know the names of. Now, I'm not going through functions here. You'll be doing that in your lecture, and I do not want to steal the fun of your online lecture. And brains are fun. It's fun to talk about brains. But the brain stem from here, see where it starts to widen, right up to here where it changes color, that's the brain stem. Medulla, pons, midbrain. Now the medulla oblongata has a name that goes with it. You'll see it in the right-hand side, the pyramids. I've never been particularly crazy about this name, but nobody asked me if I liked it or not before they gave me the name. So, if we look at the front of the medulla oblongata, so see this brain is looking directly at you. Right here is the pons, right here is the medulla. Somebody looked at this in the front and thought it looked sort of like a pyramid. I don't know that I buy that, but let me try to show it to you on a couple different models. So here's a brain model I've put together for us. Medulla and pons. The front of the medulla is called a pyramid. Now watch this nifty little trick. I do have some brains that actually come apart sort of like a puzzle or a game of, what do they call it, Jenga or something. So I can just pull out a brain stem like this. Medulla, pons. The front surface of the medulla right here is called a pyramid. Those are the pyramids. Never been real keen on that name myself, but you can see them. So that's the front or anterior surface of the medulla oblongata. If we look at it again in sagittal section, I can see medulla, pons, midbrain right there. Now you people have been in A&P long enough to know we love some goofy terms. So there's a structure associated with the pons that you must also know the name of, which could probably be seen best in this isolated, mostly brainstem. It's got some thalamus up here too, but we haven't got there yet. So let me pull this apart. And I don't mean to, beat a dead brain, but medulla, pons, midbrain right here. So now we're talking about the pons, this second bulge as we go up into the brain. And if I put this brainstem back together, I want you to notice right here and right here. Structures that look like great big giant cables, bundles of wiring. And that's only because they are giant cables, bundles of wiring, the wires here being axons. These are big tracts of wiring that run between the pons and a higher brain structure, that cerebrum. So these things are called cerebral peduncles. Look for those in the right-hand column. Cerebral peduncles, that's the weird word, peduncles, connecting the pons and the cerebrum. I have another brain stem right here. So notice everybody, the pons, and what's this? Cerebral peduncle. Cerebral peduncle. Again, sorry, but I have to jump back and forth between the right and left columns here for us to get some of these things. Now that last or superior most part of the brain stem, so once again we have the medulla oblongata, the pons, and this whole area right here, which is kind of whitish, even a little yellow there, this is the midbrain. That's the cerebellum. This is the fourth ventricle and cerebral aqueduct right there. This midbrain right here, I want you to notice it has a couple bulges back here. Let's look at a different brain. Medulla, pons, cerebellum, fourth ventricle, cerebral aqueduct, midbrain, and I see these bumps back here. These two are probably even better. A little bit weak on the medulla, pons, midbrain, but notice 1, 2 bumps back here on the midbrain. And this one might be the best in this sagittal view. Medulla oblongata, pons, cerebellum, fourth ventricle, cerebral aqueduct, I'm just repeating over and over again, and then this region right here is the midbrain. Notice these 2 bumps right here. Altogether, there would be 4, 2 on each side. So I want you to look at the right-hand column and find the term corpora quadrigemina. Corpora quadrigemina, the 4 bodies. Corp, body, quad, 4, that's 4 fingers there, sorry, I'm too zoomed in. Corpora quadrigemina. I can see this probably best, again, on one of these isolated brain stems. So look at this. On the back side of the midbrain, see the cerebral peduncle right there. There's the pons. Rotate it around. Nothing else looks like this, everyone. Do you see the 4 bodies? 1, 2, 3, 4. The top 2 are the superior colliculi. Let's say that again. Superior colliculi. The bottom 2, yes, you guessed it, inferior colliculi. And the 4 of them together are the corpora quadrigemina. This is the posterior side, the back side of the midbrain. I can see the 4 bodies best when I look at a brain stem from behind. Pay attention in your lecture to what the function of these things are. They have to do with some of our reflexes that take place in our head. Some of you will actually be testing those on people. Superior colliculi, inferior colliculi, all of them together, corpora quadrigemina. This little red thing I can see right here is not part of the corpora quadrigemina. That happens to be the pineal gland that we're going to see later. So that's the back side of the what? Brainstem. Just looking at these two brain models as examples, when somebody says brain, does that bring to mind the brainstem? The cerebellum? No. What do people think of? They think of this thing. The great big cerebrum, the big wrinkly thing. That's what this is. This is the cerebrum. That's next on our list. The cerebrum is what everybody thinks of when you say brain. Cerebrum, cerebrum. And the cerebrum has a couple fissures in it. Deep depressions. Now, I want to point out a few structures here to you just so you have the terminology right. So yes, the cortex of your brain has a lot of wrinkles in it that increase the surface area. These small wrinkles that you see here, 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 over here, each one of those is called a sulcus. I'll say that again, sulc. Think of a depression. Maybe you're sick of taking class at home, so you sulk, you're down. So each valley here is called a sulcus. The raised portions, each one, is called a gyrus. So the main area of your cerebrum has a bunch of sulci, grooves, and gyri, raised portions. So sulcus, gyrus. Looking at the other brain, right here, sulcus, the depression, gyrus, the raised portion. And then there are fissures, even deeper depressions. The first one is very easy to see, right here. This is called the longitudinal fissure of the brain, separating it into left and right hemispheres. You can argue all you want with the psychology people about left brain, right brain, and your personality traits and all that stuff. But you do have two halves of your cerebrum, and they're separated by this very nice anatomical landmark, the longitudinal fissure. All the brains have one of these, longitudinal fissure. Then we also have another fissure that separates the cerebrum from the cerebellum. This gouge right here is called the transverse fissure. Longitudinal fissure, transverse fissure. Let's look at it on the other brain. Right here, sorry for the squeaks. Longitudinal fissure, transverse fissure. Gyrus, sulcus. Terminology we use to describe the cerebrum. Okay, so you can see here I've set back up my 4 brain models on display in section, sagittal section, so we can name some of these other parts. So again, you're following along with me, I hope, in your lab manual. We've now reached one of Professor Allner's not so favorite names, the diencephalon. So listen very close to my instruction here so you know what you need to be aware of for your quiz and practical. So let's zoom in on just one of these sagittal brains, just because it's fun. Medulla, pons, midbrain, cerebral aqueduct, 4th ventricle, 3rd ventricle would be right here. Now 3rd ventricle is the cavity found there. Remember, I would have to say cavity. But there are physical neurological structures found right here as well. Now this region, from here to here, would be called by many the diencephalon, which is actually, in my humble opinion, a name best suited to the embryonic brain. So I want you to mark my words right here. In fact, hey, why don't you mark your words in the lab manual? Diencephalon will not be the answer to any quiz question or practical question I write on the brain from lab. Might be fair game for a lecture, but here's what I'm telling you. Diencephalon will not be a correct answer for any brain part in your online lab quiz. How simple is that? My reasoning here, it's very, very difficult to distinguish diencephalon from thalamus and other parts right here. So, I might put diencephalon as one of your choices, but don't pick it. In fact, this would be a good way for me to tell if anybody's watched this instructional video or not, because if somebody chooses diencephalon as the correct answer, you're just simply going to get it wrong. So I might put it on there, but it will not be the correct answer. What this area is most often called is the thalamus. This is the thalamus of your brain. I can see this very dark dot here, very circular. This is called the intermediate mass. Formerly, it would be the intermediate mass of the thalamus. This is a major relay station for incoming sensory signals that get directed to the proper place. This is sort of a router. This is your brain's version of a router right here. So this is the intermediate mass of the thalamus. This lowest part of the thalamus you see right here is called the hypothalamus. So these are different regions. Let me zoom in just a little for you. So notice my midbrain right here. So this area where the third ventricle is located, remember the cupped hands, the hands are your thalamus. This is the intermediate mass. This is the hypothalamus. The whole region is called the thalamus. Don't say diencephalon. This red structure right here in the thalamus is the pineal gland. That's the pineal gland. You will see it listed over there on the right-hand side, a little less than a quarter of the way down. That's this red structure right here, pineal gland. It's part of the thalamus. Hypothalamus, intermediate mass. The cavity again right here is called what? Third ventricle. Maybe this model makes it a little easier for us to see. I'll tell you why. First, we have the thalamus right here. Hypothalamus. This is one of the only brain models I have that actually shows this thing. Hanging straight below the hypothalamus. This will be a big deal in your Bio 224 semester. This is the pituitary gland right there. It hangs off of the hypothalamus by this little stalk right here called the infundibulum. So look down at the bottom of the left-hand column and you'll see infundibulum right there, pituitary gland right there, coming right off the hypothalamus. Remember this for 224, this thing is your blood taster right there. There's that intermediate mass again, and see in red, pineal gland. You may see it in some texts referred to as the pineal body, but pineal gland is a better term. Now, as long as I'm here, so see the top edge of the midbrain right here, corpora quadrigemina, right there. And then as I work my way up, see this thing right here, this white line? This is called the fornix. That's the roof of this thalamus. So thalamus, hypothalamus, right there at the tip of my pointer, infundibulum, pituitary gland, intermediate mass, pineal gland. Do you notice how close together all these things are? On a three-dimensional quiz, live in person, kind of tough to tell where the dot and the arrows are. On this online quiz, I can put a dot exactly where I want it. So make sure you're going through this instructional video enough times to know what it is that you're looking at. Another brain. So notice I like to use as sort of an anatomical landmark this intermediate mass, which is usually differently colored. This fornix right here, the roof. The thalamus, the depression would be the third ventricle. And the hypothalamus right there. Even easier on this thing, I think, because look at that very, very white, easy to identify fornix. This is all thalamus. This region, right where that lowercase g is the hypothalamus, the lowest part of the thalamus. Easy to see intermediate mass, and look in red, the pineal gland. Now, two other things I'll show you on this model which are very easy to identify that are in this same area, so make sure that you look for them on your list as well. So here is the fornix. Notice this white structure right here, sort of circular. This thing is called the anterior commissure. Find it in your manual. Anterior commissure. Right off the bottom front of the fornix. The anterior commissure. Again, I don't want to turn this into a lecture, but if you think about the brain in two halves, everyone, like so, these two halves of the brain communicate with each other by biochemical signals or nervous impulses. So commissural fibers, that's the technical name, would be axons, wires that run back and forth horizontally this way. So I just took a moment to prop my models all back up for us. So this structure right here, the anterior commissure, would actually be wires going in this direction as we look at it, back and forth, look at my finger, between the two cerebral hemispheres. The same with the fornix, right here. And if we move just up a little bit, right here, I can see the much larger corpus callosum. That's this thing. The corpus callosum right there. These structures, and there are others, but these, the anterior commissure, the fornix, and the corpus callosum, primarily carry signals back and forth from one side of the brain to the other. Let's look at this model right here. So I can see what? The fornix, the fornix, and the corpus callosum. Anterior commissure is not very good on this model. This model, fornix, corpus callosum, anterior commissure. In fact, what I always tell people is the fornix, right here, it forms the floor of that lateral ventricle, which would be behind this septum pellucidum. The corpus callosum with a C forms the ceiling of the lateral ventricle. Look at this model. You see this sheet coming down, which is the septum pellucidum, between the fornix and the corpus callosum. The cavity that would be behind this little wall is the lateral ventricle. Fornix is the floor, corpus callosum is the ceiling. The other structure I want to point out to you in this same view, not to be confused with the red pineal gland, is this thing right here. Blue, you see a little hint of red there. And then over here you can tell it's a blood vessel, I hope. This structure right here is called a choroid plexus. I'm going to say that again. A choroid plexus. A network of capillaries that actually leak. Fluid comes out of this capillary, not blood, similar to blood plasma, which becomes that cerebral spinal fluid that gurgles around in these cavities. So you see this red and blue right here, that's a choroid plexus. Whenever you see something like that, I'm just going to prop up another model right here, brainstem. So see the pineal gland right here? This is a choroid plexus. There's another one right there. Each of your brain's 4 ventricles has a choroid plexus in it where fluid leaks out. In case nobody ever told you this, your capillaries leak. Which ones? Yes, they all leak. But not all models show me a choroid plexus. You will notice this one does not. This one does not, but it does have that beautiful pituitary gland, doesn't it? And this one does not. But I do see fornix, corpus callosum, right? Very nice here. And a pretty decent anterior commissure right there. Again, I want you to remember in lab, you're just learning the names of these things, not the functions yet. We don't have that many structures left to know from these brain models, certainly only a couple from this sagittal section that we're looking at right here. So remember, and just by way of irritating review, medulla, pons, 4th ventricle right behind it. And remember, the 4th ventricle lives between the pons and the what? Sometimes called the small brain, the cerebellum, right here. Now, I like this particular model for the cerebellum because it very nicely shows me all of this white material here, which has the grandiose name of the arbor vitae. For those of you that speak a Latin-based language, yes, the tree of life. I can see a little more arbor vitae over here in this model. So this brain part, the whole thing, is called the cerebellum. It's got a highly dense collection of neurons here. It's a lot of gray matter. Some people think that your cerebrum up here is the only gray part of your brain. No, this is a lot of gray matter. Think about all of its functions in your lecture. But this white network that you see here, which is the axons, the wires, is the arbor vitae. Cerebellum, arbor vitae. What's the part? Cerebellum, tree of life, the arbor vitae. Same thing here. Cerebellum, arbor vitae. And last but not least, this nice model. Cerebellum, the arbor vitae, right there. The cerebellum actually has some serious mass to it, and don't take it for granted. When you learn about the functions, this thing does a lot for us. I would be very happy if I was brain part to be the cerebellum. Last but certainly not least, we have this view of the brain right here, the inferior view, where I can see structures associated with cranial nerves. Nerves that run into and out of the bottom of a person's brain. Now you have a diagram that looks pretty much exactly like this thing, both in your textbook, chapter 13 if I remember, and page 3 of this very same lab manual. What I want you to notice in your list of brain structures on page 2 is what Professor Larson has done for you in this lab manual. She's named each one of the cranial nerves and what's listed in parentheses next to it. I will wait. What's listed in parentheses next to each cranial nerve? A Roman numeral. These things are known by Roman numerals. They must be Roman numerals. But that's a big help for you, because this particular model that I have right here, you may notice, has Roman numerals on it. Duh. So in a two-dimensional online lab, we're going to take a little bit of pity on you, not a ton, but a little. So make sure you know those Roman numerals that correspond to these cranial nerves, because they're labeled here. So if you know the Roman numeral, you'll know the name of the nerve. I know some of my colleagues might object to this. You have 11 full-time anatomy instructors here at the college, but there's only one of them here. So I win. See how easy that was? So I'm going to use this model right here that actually has the Roman numerals written on it. So let's go through some of these structures. And again, we're just trying to name them. I want you to notice first, not a cranial nerve, but these two white masses right here called mammillary bodies. I'll zoom in a little bit. Mammillary bodies. They're a good anatomical landmark for us. One, they're white, they stand out. These are little clusters of neuron bodies that have to do with odoriferous reflexes and potentially memory having to do with smell. And when I look anterior to these mammillary bodies, I see two big structures. One, the two cut optic nerves. Cranial nerve number 2. The two cut, they're cut, one would go, remember the optic foramen, go to your eyeballs, right. And see where these two optic nerves meet right here in front of the mammillary bodies? That's called the optic chiasma. Find that in your list. Don't say chiasma, say chiasma. I know that's a Professor Allner thing, but it's CH. Optic chiasma, right here, mammillary bodies. These two big nerve tracts are your olfactory nerves for smell, the olfactory nerves. Notice the bulb at each end. So this structure right here is the olfactory bulb. Yours is pretty small. Other organisms, other animals have much larger ones than we do, but that's the olfactory bulb, and this is the olfactory nerve. Optic nerve, where they meet, optic chiasma, using these mammillary bodies as an anatomical landmark. But I'm not done yet with these mammillary bodies, because if I rotate this brain a little bit, can you see what's right here with the Roman numeral 3 right next to it? Look at your lab manual. What cranial nerve pair, because there's two, one here, is number 3? Oculomotor. So 1, 2, 3, olfactory, optic, oculomotor. And look at this nice view we have right here Let's jump down to the bottom of the pons. What Roman numeral is that? 6, these two. What cranial nerve pair is number 6? The abducens. Certainly. See how it works? Let's keep looking. Let's rotate this a little bit, and I'll just remove this section. Maybe we can see some things here. What's this one right here? Right there. What number is that? 7. What cranial nerve is number 7? The facial nerve, certainly. Look at this one. What number do you see? If I can get my probe in there, right there. 5. What cranial nerve pair is number 5? The trigeminal, great big giant thing. What cranial nerve pair is number 10? Right here. Vagus. The vagus nerve. And who do I have left to find? Well, let's remove a little bit here. And look at this one. What Roman numeral is that? 4. What's cranial nerve pair number 4? The trochlear nerve. So if you know the Roman numeral, 4, 5, 7, 10, 3, 6, and so on, you will know the name of the nerve, so you have to memorize those Roman numerals. I'm telling you right now. But only for those that are listed in the lab manual. What we have here, obviously, a bucket o' brains. And who does not have a bucket of brains laying around? I know that I do. You probably do too at home. And I have some brains that you can see have already been dissected, and a few, here's a nice one right here that has not. So I'm going to take this one out of the preservative, and I'll take this one right here. Nothing wrong with that one. Maybe I'll look for one more decent brain if I can spot one. Ooh, that's pretty mangled. Sometimes I don't make it too well after people work on their dissections. Let's stick with these two for now. And I can't speak for you people, but I can say for myself, I love cutting up some dead stuff. It just makes me happy. I don't know why. Like doing it. Maybe that's why I'm an A&P nerd. So what we have here are 2 sheep brains. You can see one right here, completely, totally intact, removed right out of the cranium of the animal. Here's another that's been cut in our traditional sort of sagittal section view that we like to look at. And if we notice in your lab manual, under anatomy sheep brain, I can ask you many of the same structures we had on the models of the human brain, but definitely not all, everyone, definitely not all. Let us begin with an examination of this material right here. You will hear about it in your lecture. This is the dura mater right here. Look at how thick it is. Look at how white it is. I can see it all over this brain right here. Dura mater. And yes, you say mater, not matter. The dura mater, lots of tough, strong collagen here. So this is the outermost of the 3 meninges, the dura mater. In fact, it's one that we see the best on these brains when you pull a brain out that actually has some. I can also see some pia mater, that's this shiny stuff right here. I don't think it's in our list as we go through it. But arachnoid mater, now that usually falls apart very quickly after death and dissection. So here we have dura mater, and if I set this brain down, just moving on, I can see cerebral hemispheres is the term listed. What I need to do is take this brain right here, and I will remove the dura mater for you, which takes a little bit of dissection, but I'm not above doing a little bit of dissection. Trying to cut this thing right down the longitudinal fissure or thereabouts, because that's a fairly easy thing to find externally. I can actually feel where it is with this brain. And notice I can peel back some of this very tough dura mater. See the dark material coming out? That's some arachnoid there, believe it or not. I can tell, but let's continue to do some cutting here and get that dura mater off of this thing. Okay, so you can see I've cut away and cleaned it up a little bit. That dura mater, kind of tough to get off sometimes, especially because it goes down into that transverse fissure we can see right there. So what I have right here are the two, happens to be right and left, this is the anterior side of the brain, cerebral hemispheres. The two cerebral hemispheres, here and here. And below the transverse fissure, what do I have? Cerebellum. The cerebellum right there. I can see it in this brain. Right here, notice the cerebellum with the arbor vitae, the tree of life in it, right there. On the surface of this brain, I can see what? See it, the darkly staining, because there's some blood vessels down there. Sulcuses, the sulci, and then the raised portions, the gyrus. Now, I can try to zoom in a little bit for you. So I can see very clearly the longitudinal fissure, right there. Transverse fissure, right there. All these, notice they're darker, sulci and gyri, or gyrus, sulcus. Here we go, look at this dark one. Sulcus right there, gyrus right there. Two cerebral hemispheres, transverse fissure right here, and the cerebellum right there. If I look at this brain that has been cut in sagittal section for us, let's name a few of the structures that we see here. I can notice the pons and the medulla right there. So this would be simple spinal cord. Medulla oblongata, pons, right there. And I can see above them the midbrain, right here. Now I want you to look at your list on page 5. Do you see midbrain anywhere on that list? No, you don't. So I can't ask you midbrain. I can ask you cerebellum. I can ask you medulla oblongata. I can ask you pons. But I can't ask you midbrain. I can, however, ask you the corpora quadrigemina, which is right here. Very easy to see in a brain dissection if I expand this transverse fissure. So see these 4 knobs of tissue? Get that duramater back there. Can you see all four? Perfect view right here. Superior colliculus, inferior colliculus. 1, 2, 3, 4. The corpora quadrigemina. Corpora quadrigemina. But I have to expand that transverse fissure so I'm able to see these four bodies responsible for visual and auditory reflexes. Remember that in your lecture. Here, I can tell where they are, but it's really tough to name one from the other. But what I can see very nicely in this view, look at this thing right here. Right there. That is the pineal gland. I'm going to say that again. In this brain, this right here is the pineal gland. Do you see this big circle right here? This is the intermediate mass of the thalamus. Now, intermediate mass is not on your list for the sheep brain, but that's a good indication of the thalamus. I'm in the thalamus here. So below this, right here, is what we call the hypothalamus. So hypothalamus, and then the rest of this is thalamus. This is the pineal gland right there. And notice this dark mass of stuff right here. These are actually blood vessels. That's a choroid plexus, believe it or not. Again, not on your list, but what can I see here that is on the list? Cerebellum, arbor vitae, duramater, medulla oblongata, pons, thalamus, hypothalamus. But I can also see this, so watch what I do with my hands here, everyone. Right here, watch my probe. If I cut through the septum pellucidum right here, which I just did, see that? That's a lateral ventricle. This is the fornix, the floor of the lateral ventricle. And this is the corpus callosum, the ceiling of the lateral ventricle. Fornix, lower, corpus callosum, higher. Those are part of your list. Those are things I could ask you. Also, right here is your spinal cord. Well, actually not yours, it's the sheep's spinal cord. And see this depression right in the middle of it? That is the central canal. So if we tick off some of the things on the lab manual, I've got spinal cord and central canal. I've got cerebellum and arbor vitae. I've got medulla oblongata, pons. I have the fornix and the corpus callosum. And I'm just ticking these things off the same way you will. I've got the thalamus, hypothalamus, pineal gland, the thalamus, the hypothalamus, and the pineal gland. The duramater, right here. In the full brain, we have the cerebral hemispheres, longitudinal and transverse fissure, again the cerebellum, and very nicely here the corpora quadrigemina, 1, 2, 3, 4 bodies. All very nice here in this view, and as well as a whole bunch of duramater. I have very nicely shown here sulci, a sulcus, the depression, gyrus, the raised portion, and this just for lecture. Notice that there's a thin tissue covering this cerebrum right here. I'm under it right now like saran wrap. That's the pia mater. only for your lecture, but that's the pia mater. Some of the other structures I'm going to need to clean up and look at the underside of this brain, so I will do that. Okay, everyone, I had to give up on that first brain I was working on to see the underside because there was a major hemorrhage here where the pituitary gland is. How could I tell? I don't know. Something gave it away. Look at that. Yeah, I'm the sheep medical examiner here. So I just randomly selected another brain and dissected it out, took off its dura mater. So a couple prominent features I can see on the bottom of this brain. This structure right here, which is the pituitary gland, directly behind, look at this everybody, optic nerves, optic chiasma, right there, so right behind it is the pituitary gland. So I've got optic chiasma right there, pituitary gland right here, and then watch what happens if I fold these optic nerves down. Now I can see these two sort of white structures right here. These are the olfactory bulbs. And then the nerve that runs back from them, I can see this one right here, is the olfactory tract. Notice how much larger these are as a portion of the brain than yours or mine. This sheep has a much better sense of smell than we would have. But the cerebrum isn't that big as a part of its brain, so it's probably not doing any calculus. If we notice this top portion of the cerebrum, which is the cerebral cortex, can you see how it's a little bit darker than the underlying tissue? So right here, this top on this sheep, it's probably the top, I would say, 1/8 to 1/4 of an inch of the cerebrum is the cerebral cortex. So I've got all the structures hit here except for one, the cerebral peduncles. Do you remember how to find one of those? Here's the pons. Look at the front side of it, pons, and then this tract coming right out of it right here is the cerebral peduncle. Let's see if I can show it to you on this brain after I do a little bit of dissection here in the pons area. I've dissected this brain in such a way now that you can see the pituitary gland right there, but I can also lift it up a little bit. So see these two tracts coming out of the pons, cerebral peduncles, everyone. This again is that pituitary gland, kind of rare to find in a lot of these brains because when the brain is removed from the skull, the pituitary gland actually hooks on that sella turcica lots of times and gets ripped right off. So make sure you're going through these structures in this instructional section and in the practice video. Because it's brains, and dead brains are just cool.